All right. Note to self: do not turn on drawing tools. Um, I had to restart to get rid of that junk. Okay, so again, cutting edge embryonic stem cells. Question at the end of the chapter. Definitely pay attention to that. To this, that this is the stereotypical picture of the average neuron. We've already seen that all neurons don't look like this. This would be called a multipolar neuron because it has multiple poles and lots of dendrites, and it looks like flattened roadkill, but it's three-dimensional, and these dendrites are probably bristling with synaptic spines, very complex. And everywhere where there's a synapse, by the way, information coming into the cell, it often has the option of talking back. It's bidirectional. Now, in this picture, the myelin is not part of the neuron. It's a common misconception that it is, and figures like this reinforce that. The text will straighten it out. But the myelin is produced by glial cells. They are separate from the neuron, and as you'll see in the chapter, very relevant to MS. I had Guillain-Barré in high school, where my myelin was attacked by my immune system, not in the central nervous system where MS happens, but in the peripheral nervous system. That was kind of fun. So, to oversimplify, this is the receiving end of the neuron, but we know that these dendrites can talk back. Decisions are made at the axon hillock whether to convey the action potential down the axon. I like to think of the action potential as being like a burning fuse. And if a burning fuse got to this junction, which way would it go? And of course, the answer is it goes both ways. And then it goes here. And when it gets to junctions, which way does it go? And it goes all ways. So it's complicated. So there's, discussions of dendrites, and there are, thousands of them potentially on a cell, and their influences interact. I went looking for pictures of spines on neurons, and I can't beat this one. It's just bristling with spines. Uh, and you hopefully you can recognize these as pyramidal cells. Okay. And this is one form of myelin. Notice that there's a glial cell nucleus here in this wrap. That looks like some food. I forget what food, uh, but Pillsbury sells these wraps you can put around hot dogs. Um, but if there's a glial cell there, that means this is a Schwann cell, and that will be defined in a bit. And Schwann should be capitalized, like Purkinje. And these are the cells that were attacked in me when I had Guillain-Barré. They provide myelin in the peripheral nervous system, and they actually help axons regrow if they are damaged. This is a nice cross section between two segments of myelin. Notice that there's a gap, node of Ranvier. That's extremely important to action potential propagation, as we'll see. And here's a discussion of the axon hillock and axon collaterals, terminal buttons or boutons, because the French used to be important, and another indication of the importance of France, nodes of Ranvier. Okay. And then the soma, runs the show largely, but it's, the phrase was and sort of is, all roads in the Roman Empire lead to Rome. The roads were not unidirectional. They go both ways. And in the video I shared with you, you're going to see little guys transporting things through the cell. It's just amazing. When I teach about genes and protein synthesis, I simply say DNA, RNA, protein synthesis. This is a bit more advanced. Take a look at it. It's important. These terms are important. Epigenetics, boy, is this a hot topic right now, but be warned, there's really good science behind epigenetics, and even epigenetics going transgenerational, where what happens to a grandmother or grandfather can affect grandchildren. That's all real, but there's a lot of flaky people out there hooking on to epigenetics, imagining that we are basically a blank slate upon which you can write anything. Don't adopt that. So epigenetics from a scientific resource, very good, but epigenetics from these crystal people and getting in synchrony with the vibrations of the universe, no. So that's worth looking at. It's very important. One of my research colleagues, when I did mice research, was very into this. It's definitely real. Look at all a whole other page on it. It's very important to modern neuroscience. So this is like a cinnamon or a gingerbread man version of the neuron. The purple thing over here is the nucleus, and then here's the soma or cell body. And then I talked about the transport mechanisms, which really don't look like that, but there is transport down the axon to the terminals and from the terminals back to the soma. So, the Romans wanted to get their stuff, particularly troops, out fast if they needed them. And they also wanted to ship stuff they produced. But also, they would like to know what's going on out in the colonies. So there are mechanisms that snack on the environment of the terminal, and it's shipped back, and that can interact with the DNA potentially of the nucleus, and DNA, RNA, protein synthesis, and epigenetics and everything. There's a discussion of the axoplasmic transport, and again, the amazing little guy pulling the vesicle in the video I shared. It's real. Now we're talking about the cell membrane. Now we mentioned the cell membrane already, bilipid layer. Again, if you look at the video and you look for the bilipid layer, it's floaty, and there's all these little round heads on it. Those are the hydrophilic ends that are both on the inside and outside of the cell. But there are transmembrane proteins that allow things to move across the membrane that are not lipid soluble. And now we're getting into sodium, potassium, chloride, and calcium. Sodium and potassium we spend most of our time with, but calcium is critical for the release of neurotransmitters. I did a lot of research on morphine, and remember the drug I mentioned, naloxone, which is Narcan. The calcium is really important for morphine to work. And I injected chemicals into the brain, intracerebral, which you'll remember from the last chapter, which means directly into the brain. And if you put things that bind calcium into the brain and then inject morphine, not much happens because without the calcium, the morphine's influence on the cell is blunted. So that's all important. And now we're revisiting receptors. There was a very simple view of receptors in the last chapter. Here we get more mature. There's again a nice picture of the bilipid membrane with the round balls on the outside and the inside, and the squiggly things that are hydrophobic in the middle, and these are transmembrane proteins. They're proteins, and thus they're proteins, and they're transmembrane because they go across the membrane. And it's not one giant protein; it's usually 5 or 6 that stand together. And by the way, in the videos, you saw some proteins getting together and standing together. And if they stand together, transmembrane, they can produce a channel or a pore, which could be closed, as is the case here, or it could be open, as is the case here. Ligands were mentioned in the first chapter. Here's a ligand binding with, notice, by the way, the receptor is a tiny part of this receptor complex. You wouldn't believe, but by the end of the course you will, how complicated these things are. It's like Star City from Star Wars. You can land on many sites, and the different sites can have different functions, and they can even fight each other. Very complicated. But we're going slowly now. So the ligand can go here, and in this case, it can open the channel. And of course, if you would put a receptor antagonist there, the ligand couldn't, the endogenous neurotransmitter couldn't bind, and therefore the channel wouldn't open. And this is a little more complicated, but here we have something on the inside of the cell tickling the feet of the transmembrane proteins, and by interacting with the inside of the cell, you can also change whether the channel is open. And in the middle, we have voltage-dependent channels. You don't need a ligand. You just need an electrical potential, and they respond to that. Confusing now, but it will become less confusing, hopefully in a bit. There's the glial cells again. Here's the Schwann cell. Notice that it will have a cell body, peripheral nervous system. Here are oligodendrocytes or oligodendroglia. Notice that the cell body is separate and they glom onto multiple axons. These are the ones that are being destroyed in MS, which is a central phenomenon. And this is what my immune system attacked when I had Guillain-Barré. Astrocytes, the glia, again, if you ought to invest money in the future of the nervous system, I would invest in glial cells. Very important. And consider investing in gut microbiome. So you read that and you appreciate it. And here's a box that overviews the astrocytes, the microglia, Schwann cells, oligodendroglia or oligodendrocytes, and what they do for a living and where you find them. Very nicely done. Can't improve on that. And here we go into electricity. You can't understand the action potential unless you understand the resting potential. And the resting potential is what you get almost automatically, although we do have pumps, but don't overthink the pumps. I like to think of the membrane being polarized -70 inside relative to out. The book does too. That's good. Some books give different numbers, and I'm always contradicting them. It's not important what the exact number is, but -70 is what I like to say. Now, -70 thousandths of a volt, millivolt, is not a lot of volts, but I'll tell you, that amount of potential difference in a structure as small as an axon, that is killer. And this potential can jump from -70 to upwards of, +40, +50, like an over 100 millivolt shift in potential. That's big. And if it weren't big, we wouldn't be talking. Okay, so here's an example of how you figure that out. Here you have two microelectrodes. Well, they're structurally different. One is indicating that it's the reference electrode, and one is the recording electrode. I'm not sure I would have drawn them that way. When they're both outside the cell, it's sort of like, they're in an ocean, they're in salt water. And if they're in the ocean in the same glass of salt water, there's not going to be an electrical potential between them. But the bilipid membrane of the cell, or the axon in this case, which is part of the cell, you drive this puppy into the axon, which, by the way, is quite a trick. Welcome to the Nobel Prize. And suddenly you go from no potential to a step-like function of roughly minus 70. So this is a giant squid axon, not an axon from a giant squid, but from a giant axon in a squid. Stay calm with this table. You can refer back to it from as you read the text. So on top, you have concentration in moles outside and inside. And all you really have to know is there's a pot full of sodium outside the cell and not so much inside the cell. And then, oh, look, potassium, it's the opposite. A pot full inside, not too much outside. Chloride is definitely important, and it has more outside than in. Calcium, as I said, is very important for neurotransmitter release. And look at how this puppy is set up. The outside has a bunch, the inside has like nothing. So if calcium can move from outside to inside the cell, stuff happens. Proteins, if they start moving across your cell membrane, that cell is probably dead. They're not going anywhere. They're important to the overall story, but they're trapped. There's no channels for proteins. Basically, only if the cell membrane is ripped asunder do they get to move. You're going to get confused about potassium. Potassium has pores or channels that are always open, which means potassium can move across the cell membrane pretty easily almost all the time. I must admit there are extra potassium channels that you will fixate on to the point of getting confused later that open during the action potential that make it easier for potassium to move, but potassium would have moved anyway. Sodium, the channels by and large are pretty much closed, and so potassium is not going to be able to go through these. And you're going to have to read the text to go with this one. But this is your sodium-potassium pump, and it's trying to keep things unbalanced. It is pumping sodium where there's too much of it out, and it's pumping potassium where, in a sense, there's too much of it in. It is very expensive metabolically, and we're not doing it for nothing. But when you think about the action potential, fight the urge to think about this thing turning on and off. What happens is largely channel driven. Electrostatic pressure is driven by electricity. Who would have thought? Concentration gradient is based on concentration. These words were not invented to be confusing. Have faith. Okay, equilibrium potential is that potential where an ion species is going to maintain a stable intracellular and extracellular concentration. The cell is almost at the equilibrium potential for potassium. It's not there, almost. It is crazy off for sodium. Really off. Ooh, can you name the morphology of this cell? It's our old friend, the pyramidal cell. And there's a nice graphic there showing, we go into the cell and we generate these little potentials that don't reach thresholds, or no, these are hyperpolarizing. I would call this hypopolarizing, but the book likes the term depolarizing. It's common to say depolarizing, but I like hypo. And this little red dashed line, that's your threshold of activation. And if your membrane can drift from its resting potential of minus 70 to what they're saying here, minus 50, I usually do minus 60, all hell breaks loose. And the voltage dependent sodium channels, by and large, open, and all that sodium, it's like Black Friday at Walmart. They open those doors, and if you don't get out of the way, they kill you. And all the sodium doesn't come in, only the sodium near the doors, because in very short order, you get to the peak of the action potential, and the doors close, and the cell is not filled with sodium that has to be pumped out. It's like you open the doors by accident to Walmart for 5 seconds. A bunch of people get into Walmart, and then they say, oh my God, close the doors, it's too soon. What do the shoppers do? They walk into Walmart. They move away from the bilipid membrane. They do not fill Walmart. Just keep practicing that. And the falling phase of the action potential is potassium. And potassium's fall is facilitated, not caused by, but facilitated by the extra potassium channels that open during the action potential. And now we're showing how things can interact. Remember, there are thousands and thousands of synapses, and it's like a sort of our democracy. We get to vote out here in Scranton, but the people with real power, the lobbyists, they're in the Capitol, and they are lobbying, and their influence on whether the action potential fires is way greater than us. Sadly, it's a lot like democracy. Sorry for getting political. So let this wash over you. It talks about sodium and potassium. Notice this hyperpolarization after the action potential. See, we started at minus 70, and now we go down to like minus 80 or so, minus 83. That's because of the extra potassium channels that opened. Extra. And because you're down here instead of here, to get to threshold, you used to have to go up 20. Now you have to go up maybe 30. So this is very important, and I'm kind of obsessed with it. And here comes France again. So this is a multipolar neuron. It has an axon. The axon, by the way, may be in part in the central nervous system and in part in the peripheral nervous system, which would mean that the myelin in the central nervous system is oligodendrocytes, and then once it gets outside, it'll be Schwann cells. They have the little nodes of Ranvier. Notice that's the only place we have pumps. So myelin increases speed, but it also saves a lot of energy, because under the myelin, we don't have to pump. And by the way, if you get Guillain-Barré or MS, and this myelin dies, you have a whole segment of messed up axon because you don't have pumps there. And often it doesn't just slow things down, it stops. That table is good. That's good. Okay. I usually lecture about this using food. So if you're doing a coronal section, the bottom one here, you're making sliced bread. You hold the bread down on the table, and slice it straight through, coronal, cross section. If you're making a hoagie, at least as we make them in this area of the country, you hold the bread down, you put the knife parallel to the countertop, and you slice horizontally. Sagittal is a weird one, but I spent many happy years scooping ice cream, and I loved to make banana splits. And to make a banana split, you take your banana, hold it screaming on the table, and hold the knife perpendicular to the table and split the banana. You serve me a banana split where you have slices like sliced bread, I'll send it back. That's not split. Those are coronal sections. And then there's the planes of section, subdivisions of the nervous system. This is a brilliant figure here. A lot of textbooks get this wrong. Notice where the spinal cord ends. There's the spinal cord. It's ending at the upper curvature of your lower back. Down here, there is no spinal cord. You will find textbooks that have you sitting on your spinal cord. Wrong, wrong, wrong. So you have central and you have peripheral, and there's all those subdivisions, and that's all fun. I'm a pain guy, and here we have the spinal cord, and I hope you can look at this and figure out what kind of section it is. It's a cross section because you took your spinal cord and you held it down on the table and you cut across it. Here we have the dorsal root ganglion, because this is the dorsal root. There is no ventral root ganglion, only a dorsal root ganglion. And the cells in there really look like that, like little balloons on sticks. And they have a little stalk, and one branch goes out to your skin often, and the other branch goes into your central nervous system, which is your spinal cord. So this guy would have Schwann cells out here, and they would switch to oligodendrocytes in here. And then in here it gets complicated. We've got interneurons, and it's complicated. And here are your motor neurons. And again, you'd have oligodendrocytes here and Schwann's out here. Notice that things come out of the spinal cord pretty, spread out, and yet they gather together in these mixed nerves and the dorsal ganglion stuff. Why? Because we have vertebrae, and the vertebrae are stacked, and to get out, you go between them. So each segment of your vertebrae has one of these skipping out. Autonomic, for some reason, it helps me to say that the sympathetic nervous system isn't. It winds you up, and too much of it can be destructive. If you're digesting lunch, that's parasympathetic. And I alluded to drug-dependent, state-dependent memory in the last chapter. If you are calm all the time when you study, that's this state. And then you take an exam and you get all stressed out and you turn stupid. That's because your sympathetic nervous system has gone crazy. So there's a nice table that overviews that. This figure has a bunch of stuff. Certainly you won't be memorizing it, but notice that there's norepinephrine in here and acetylcholine. Noradrenergic, norepinephrine, cholinergic, acetylcholine, and we have pre and post-ganglionic. So let's look at your stomach. You have blue pre-ganglionic, cholinergic going out to ganglia. They talk in the ganglia to a second cell, and that comes out and uses norepinephrine there. Sometimes things go directly to the organ, and there are lawful differences between sympathetic and parasympathetic. Nuclei. Now, when you think about single cells, you think about the nucleus, but a collection of neural cell bodies in the central nervous system is often referred to as the nucleus. Collection of axons in the central nervous system is often referred to as a tract. In the peripheral nervous system, a collection of cell bodies is often referred to as a ganglion or ganglia, and axons are called nerves. Oh boy, neurotrophic. This is very big, very hot topic. I'll leave it to you to look at it. You really, really, and if you were in my regular class, I would test you until your ears bled. You really have to learn this table. You have to know it cold. The reason you have to know it cold is these terms are going to come up over and over in the course. So spend some time, do some brute memorization, learn that table. By the way, I like brainstem to be 2 words, but I'm old. There's a beautiful picture of your spinal cord and how things are skipping out between your vertebrae. There's a cross section of the spinal cord showing lots of tracks. Know the table cold, then these terms won't surprise you. Locus ceruleus. There's a color that should come to mind if you like color in painting. Cerulean blue. The locus ceruleus is called the locus ceruleus because in fresh tissue, it often looks blue. I spent a lot of time working around the dorsal raphe nucleus. So the locus ceruleus, norepinephrine. Raphe nuclei, serotonin. If you learn those things, it will simplify the rest of the text. This is not cut. This is a mid-sagittal cut. The cerebellum, which is here, I like to think of it as a piece of cauliflower. It is not part of the brainstem. It sticks on the brainstem. It's not part of the brainstem. Mesencephalon or midbrain, I spent a ton of time doing research on that in rats, and we'll see rats in a bit. Basal ganglia, Parkinson's disease, and other things. The periaqueductal gray, that was my life for a long time. Periaqueductal means around the aqueduct. And as I mentioned, in Roman times, aqueducts carried liquid. And the cerebral aqueduct carries liquid from the 3rd to 4th ventricle. And there's gray area around it, and it's called the around the aqueduct gray. Very important to me. Substantia nigra. That refers to black. And the substantia nigra in a fresh brain, unless you have Parkinson's, is very obvious and pretty black. The ventral tegmental area, or more mercifully VTA, that's going to come up over and over. It's involved in reward and addiction. You have to know the VTA, reward and addiction. And another area will come up later that connects to that. And now we're into the diencephalon, 2 major structures. If you are hypothermic, what is your body temperature? It's low. And the hypothalamus is below the thalamus. And it's small, but oh my God, it's action-packed. And there's a figure coming up. Here we go. Geez, look at all the stuff in the hypothalamus. Many of that will be important. Don't try to memorize it, but much of that will be important later. And here's the master gland. It's more than one gland, the pituitary down here. Limbic system, we're going to spend a lot of time on that, a lot of time. And I'm trying to say it's not labeled, so I'll skip it. The amygdala is going to be big. The hippocampus, where there are pyramidal cells, that will be big. This is the stress story. I've done research on the ACTH. Take a look at that. Lobes of the cerebral cortex. We have pretty giant frontal lobes. pretty giant. And corpus callosum, the largest bundle of myelinated fibers in the central nervous system. This is what we cut primarily to produce what's called a split brain, where you disconnect the left and right lobes. By the way, again, thinking about food, when I look at the cerebral cortex, I think about walnuts. Looks like a walnut. And down here, that's cauliflower. And there's, lots of parts, lots of things to know. Okay, here's us versus rats. And I did most of my research in rats. I think they have shortchanged. See the midbrain up here, purple? They shortchanged the rat on the midbrain. It really should end about here. The pond should be bigger, and they should have moved the medulla up a bit. But look at the olfactory bulbs of rats. They're not even almost visible in humans. Rats really have it going on in relation to olfaction, and African rats have been trained to sniff out tuberculosis and landmines. And again, a comparison between humans and rats. And we're done. How's my time? Nice. So let's look at the study questions. Embryonic stem cells, I didn't talk about them. They're hot. They're sometimes controversial. We're growing little brains out of them now, three-dimensional brains. And there's a very extensive discussion of them there. Major features of the neuron and what they do for a living, that's like bonehead very easily. What is myelin? Why is it important? Don't forget that there's two kinds of cells that produce it. Protein synthesis, which I did shortchange, but the book doesn't. And epigenetic, which is so hot. You really should understand epigenetics, but don't get sucked into the alternative universe of crystals and stuff. Cytoskeleton, normal exoplasmic transport. I didn't mention Alzheimer's, but that's important. I really recommend, but don't require, that you look at the video because you will see exoplasmic transport. Ligand-gated and voltage-gated ion channels, really important, particularly in a chapter on action potentials. There's that table on the 4 types of glial cells. This is a growing area in neuroscience. You really should be conversational in glial cells. And then, what is the resting membrane potential? And I like -70. What makes it happen? Make sure to describe the establishment of the equilibrium potential for potassium. Again, we're close to it, but not exactly at it. The membrane potential of a cell is defined primarily by ions that can move. Because you were probably wondering, well, I've always heard about sodium, and there's all the sodium. Why isn't it affecting the membrane potential? Because it can't move. But potassium can. So at rest, you're looking at a potassium membrane. But when the Walmart workers open the doors on Black Friday, bam, sodium comes rushing into that cell, and it goes from being because sodium is so out of whack in terms of where it should be, it doesn't flood the cell, but it sends a lot of ions across the cell membrane. And briefly, the membrane goes from being a potassium membrane to a sodium membrane. But then, as you'll see in the chapter, the sodium channels close. And frustrated shoppers are out in the parking lot. And the shoppers that got into Walmart just happily go shopping. And then potassium finds itself in a very weird place. It was happy at -70. And now it's like +40. So potassium starts dashing out of the cell. and extra channels open, which is why it hyperpolarizes. Um, it's pretty elegant. What happens at the axon hillock? Movement of ions. What causes the absolute refractory period? That's sodium channels deciding to shut up. And the relative, that's the one that's confusing. That's the one that's confusing. And if you want to test yourself on whether you understand potassium, if I gave a drug to an animal and made it so that the voltage dependent, only the voltage dependent, remember potassium can always move, only the voltage dependent potassium channels can't open, what would happen to the shape of the action potential? And if you think it's going to stay up flat at the top, you are mistaken, because potassium could always move at the resting potential without those channels. So just give that some thought. I won't test on it, I don't think, but you never know. Local potentials, action potentials, that's pretty easy. What is a somatic nervous system? It's various parts. And don't forget about the difference between the dorsal root, which has dorsal root ganglion, but the ventral root does not. Autonomic nervous system, sympathetic and parasympathetic. You don't have to go into crazy details on that, but just generally, four protective features of the CNS: skull, meninges, cerebrospinal fluid, and blood-brain barrier. Um, I'm a big fan of CSF. The brain would be in total trouble if it weren't floating in salt water. There was a woman who got a spinal tap and they didn't detect that her CSF pressure was to the moon. And they stuck a needle in her lower spinal cord and CSF came blown out of there. And her brain got sucked down into the hole at the bottom of the skull and sucked the brainstem and the blood vessels associated with the brainstem into that hole. She was so dead. Hopefully, if you ever get CSF taken, they'll check your pressure because there's other ways to get it. Neurotrophic factors, I didn't discuss that very much, but the book did. The basic functions and cell groups in the spinal cord, medulla, pons, and cerebellum. The book, by the way, in humans is putting my area, the periaqueductal gray, in the human pons. A lot of it. I hate it that the mesencephalon isn't even mentioned. Good. PAG, substantia nigra, ventral tegmental area. How do the latter two nuclei control dopamine function? Right. Substantia nigra and ventral tegmental both have cell bodies that produce dopamine. Hypothalamus, does everything. Limbic system, there are parts. What does it do? Cerebral cortex, rat and human. There are those figures. Let me just quickly show my former life. And this is the close. This is something I did in 1982. What were you up to in 1982? Evidence for opioid, and this is appropriate use of the term because it's the brain's own chemicals, and non-opioid forms of stimulation produced analgesia in the rat. So we looked at SPA from midline regions in the midline. Dorsal placements were in the caudal PAG, periaqueductal gray. Ventral were within or below the dorsal raphe, and raphe are important. SPA thresholds were consistently higher. We don't care about that. SPA from both dorsal and ventral differed in the following ways. One, post-stimulation analgesia, that is when you turn it off, how long did it last, was more difficult to obtain in ventral than dorsal. During stimulation, analgesia did not vary, although continuous description of thresholds was seen for ventral. Thresholds that is, if you graphed the thresholds, they were just like a shotgun. They were just all over the place. But for the dorsal, there were two clusters. We never did figure that out. And what's most important in naloxone, and I said, which is also Narcan, reliably elevated SPA for the ventral but not dorsal placements. We conclude that different substrates of stimulation-produced analgesia lie in the close proximity to one another in the medial midbrain of the rat. It seems to have both opioid and non-opioid. And 20-some years later, they figured out the non-opioid was endocannabinoid, marijuana. Ask me if I'm frustrated that it took 20 years to figure that out. Okay, thanks for your attention. And that's the end of chapter 2.